Hello everyone, and in this video we're going to be talking about natural killer cells and cytotoxic T cells. We're going to be starting off by talking about cytotoxic T cells, and we're going to be talking about how cytotoxic T cells as well as natural killer cells recognize what to kill and how they actually kill an infected cell. Now let's start off by talking about cytotoxic T cells. They're going to recognize what to kill through the MHC class 1 molecule on the surface of the abnormal cell. Each of our nucleated cells are going to have a MHC class 1 molecule on their surface. And an abnormal cell can present an antigen on the surface of their MHC class 1 molecule. So here is an MHC1 molecule, and the circle is going to represent an antigen. And the way cytotoxic T cells, also known as CD8 positive T cells, recognize these is through their T cell receptor, which I'm drawing here, as well as the protein CD8. So that is why they're named CD8 positive T cells, is because they have the protein CD8 on their surface, and this protein is going to help them recognize what to kill. Now that the cytotoxic T cell has recognized what to kill, how does it actually kill that abnormal cell? And the way it does it is through two ways. The first way is through the release of perforins and granzymes. So the green circles that I'm drawing are going to represent perforins. The blue circles that I'm drawing are going to represent granzymes. So these perforins are going to create pores on the abnormal cell. Then these granzymes can then enter that abnormal cell and induce apoptosis. So that is one way in which CD8 positive T cells kill abnormal cells, through perforins and granzymes. The second way is through the interaction between Fas and Fas ligand. So on the surface of the abnormal cell is going to be Fas. Let's label this here. And then on the surface of the CD8 positive T cell is Fas ligand. When they interact together, it is going to induce apoptosis of the abnormal cell. Now let's talk about natural killer cells. Natural killer cells kill infected cells the same way as cytotoxic T cells, so that is why their functions are very similar. They are also going to release perforins and granzymes, and they're also going to have the Fas ligand on their surface. And when they interact with the Fas on the surface of the abnormal cell, they're going to induce apoptosis of that cell. So they're very similar. But the one notable difference is how they recognize the infected cell. So let's write this down, how they recognize the infected cell. There are going to be three ways in which natural killer cells recognize abnormal cells. First is the lack of MHC, and this MHC1. This can happen with cancer cells. As I said previously, all nucleated cells in our body have a MHC1 molecule on their surface. If there is not an MHC class 1 molecule on the surface of a cell, then this could be indicative of a foreign cell. So here, natural killer cells, they have something called a inhibitory receptor on their surface, and the MHC1 molecule is going to So they have something called the inhibitory receptor on their surface, and the MHC1 molecule is going to bind to that inhibitory receptor, and it is going to inhibit the natural killer cell from killing that cell, okay? If a cell does not have that MHC1 molecule, then the inhibitory receptor won't be inhibited, and the natural killer cell will be active and could kill that cell. Now, the second way in which natural killer cells recognize what to kill is going to be through the presence of surface proteins or carbohydrates, through the presence of foreign surface proteins and carbohydrates. So let's also write this down. So presence of foreign surface proteins and carbohydrates. So natural killer cells are also going to have a receptor called the activating receptor on their surface. With the presence of a foreign protein or a carbohydrate, it can bind to that activating receptor and activate the natural killer cell to kill that infected cell. The last way in which natural killer cells recognize what to kill is through something called antibody dependent cell mediated cytotoxicity. This is also abbreviated as ADCC. So this is a fancy way of saying that if an antibody is attached to something, then this is going to induce the natural killer cell to kill that cell. So essentially, antibodies attach to anything that's foreign. As a result, if an antibody is attached to something, then natural killer cell can come and kill that thing. So here on the surface, let's erase this portion. So let's say that this is a IgG antibody. The Faz region is going to be binding to the infected cell, and the Fc region is going to bind to the Fc receptor that is on the surface of the natural killer cell. It is important to know that the Fc receptor is also called CD16. A unique protein on the surface of the natural killer cell is going to be CD56. So let's write this down too. CD56 is going to be indicative of a natural killer cell. And as we mentioned earlier, once the natural killer cell recognizes the infected cell, it is going to kill the infected cell the same way as a cytotoxic T cell will, as we discussed previously, through perforins and granzymes and the interaction between Faz and Faz ligand. Now, contrast the way natural killer cells and cytotoxic T cells recognize what to kill. With cytotoxic T cells, it involved MHC1, so the antigen had to be nicely presented on the MHC class 1 molecule. With natural killer cells, it was different. So we talked about the 3 ways in how they recognize the infected cells. Now let's talk about how natural killer cells are stimulated. they're going to be stimulated by IFN alpha, IFN beta, interleukin 2, and interleukin 12. I like to group IFN alpha and IFN beta together because these are going to be released by the infected cell. So it is basically a way of the infected cell to say, hey, I'm infected, I've been invaded, come and help me, come and kill me. Okay, so come on, kill me is how I would remember IFN alpha and IFN beta. We can group these together. When an infected cell wants to be killed because it's been infected or invaded, then it is going to secrete these. And that way, the natural killer cell knows, hey, there's something going on, let me come and do my job. Now, IL-2 is going to be released by T helper 1 cells as well as natural killer cells. And IL-12 is going to be released by macrophages. So what natural killer cells excrete, so the cytokines secreted by the natural killer cells are going to be IL-2, interleukin 2, and IFN gamma. So the way you can think about IL-2 is it is basically a way for the natural killer cell to perpetuate its own activity. So interleukin 2 is going to stimulate the natural killer cell, and in return, natural killer cell is going to release the cytokine interleukin 2 so that it can perpetuate its own activity. So it's pretty cool. Now the surface proteins that we've talked about, let's mention these again. Surface proteins of natural killer cells are going to be CD56. This is the unique protein on the surface of natural killer cells. CD16. Remember, this is the Fc receptor that is going to bind to the antibody. That way, the natural killer cell can recognize what to kill by binding to that antibody. And lastly, we talked about the Faz ligand, which binds to Faz, and then this is going to induce apoptosis. And this is all you need to know about natural killer cells and cytotoxic T cells.